We'll go to our last speaker, who is Elektra Schubert. And Elektra is from the University of Melbourne, and she is going to tell us about decoding explicit and implicit representations of food, health, and taste attributes in the human brain. So whenever you're ready, Elektra, take it away.
Thank you. Can everybody see that? Does that work?
Yeah. Yep, that looks good. And I'll ask you to speak up a little tiny bit because you're a little soft.
Okay, no worries. So hi, everyone. Thanks for tuning in. My talk is on decoding representations of food, health, and taste attributes in the human brain. So as you're probably aware, there's a global obesity health crisis, and excess body weight has all kinds of other health problems that it's associated with, including heart disease, high blood pressure, type 2 diabetes, and various cancers. So part of the cause of this is poor food choices. So it all kind of comes down to this situation in the supermarket or the convenience store where we're faced with all kinds of food cues. So we might see a Kit Kat packet, we might see a popcorn packet, and it's important to understand how people process these food cues in order to understand how we make food choices and how we might improve these food choices. So when we make food choices, there are two main attributes that go into these decisions. The first of these is taste, which is a hedonic and fairly low-level representation. So it's thought to be integrated into decisions fairly quickly and automatically. The second attribute is health, which is a more abstract and high-level representation, and it's thought to be integrated into food choices more slowly. So because these attributes are often processed fairly quickly and automatically, it's useful to use neuroimaging methods such as EEG to understand how they occur. And a couple of studies have done this, including this one by Schwab et al. They looked at an ERP component called the late positive potential, which occurs around 450 to 1000 milliseconds after a stimulus or a food image occurs. And they found that it was more positive in amplitude when people viewed images of sweets and meat compared to when they viewed vegetables. So on the graph here, if we compare the red and blue lines, which were the sweets and meat, to the green lines, we can see that the more high calorie foods had a higher LPP amplitude associated with them. So they suggested that the LPP might reflect the processing of high versus low calorie foods. And in a recent study in our lab, we found kind of a similar thing. So we looked at differences in LPP amplitude between sugary drinks that people had rated as tasty compared to drinks they had rated as not so tasty. And similarly, we found that the LPP was higher in amplitude for the high taste drinks compared to the drinks that they rated as less tasty. So it seems like the LPP reflects the taste value or caloric value of foods and drinks. And it seems that we might have information at a very early stage in the brain about the attribute of taste, but it's not so clear whether people also process health quite as quickly. So that's why in our study, we decided to conduct two experiments to see whether it was possible to decode both of these attributes from brain activity during the first second of people viewing a food cue. So in experiment one, we had 37 participants, and they viewed 174 images of foods from this FoodPix database. And here are some example images down here. We chose the images so that they varied a lot in both tastiness and healthiness. So there was representation from lots of different food categories. The task itself was quite simple. So this is basically how it worked. They would view a food image for 2 seconds, and then they would rate on this continuous sliding scale either how much they enjoy the taste of the food or how healthy they consider the food to be. And each image was presented twice, so once for each of the two questions, which resulted in 348 trials. And this occurred in 12 blocks. And within each of these 12 blocks, only one of the two questions was presented. So within one block, they would only be rating either how tasty or how healthy they considered each food to be. And during the task, EEG was recorded. And this is how we analyze the EEG data. So we used a technique called MBPA, which Tyle talked about in his talk already. But our approach differs a little bit in that we used support vector regression instead of classification. So this involves training an algorithm on a set of data to use these patterns that are distributed across multiple electrodes to predict, in our study, the health and taste ratings. And its performance is then tested on the remaining data. So I'll kind of walk you through what this image is describing here. So we had a 20 millisecond window that moved through the EEG data in overlapping steps of 10 milliseconds. And these windows were then transformed into spatiotemporal vectors. And we also had vectors of the taste and health ratings for each trial. We then trained an algorithm on 90% of the data to predict the taste and health ratings based on these vectors of the brain activity. And then its ability to predict the taste and health ratings was tested on the remaining 10% of the data. And then what this gives us is a graph that looks like this. So we have time on the x-axis, and then on the y-axis, we have the correlation between the real labels, so the real taste and health ratings, and the taste and health ratings that were predicted by the algorithm. And then the blue line here is the real decoding, so it was the algorithm's performance. And then the yellow line here is a chance distribution. So it's basically the same analysis that I described in the previous slide, but with all of the taste and health ratings randomly shuffled. So then to compare the classifier's performance to chance, a t-test is performed at each time point, and we have significant time points highlighted in purple, pink here. So we can see that for the taste decoding results, it was possible to decode taste based on the brain activity from a little bit after 500 milliseconds. And this seems to last as a fairly sustained representation up until almost 1000 milliseconds. So then we can compare that to the results for health, which showed a weaker and kind of later decoding onset. So decoding was possible from a little bit after 600 milliseconds instead, and it doesn't seem to be anywhere near as strong or sustained as the results for taste were in the previous slide. So based on this, we decided to reanalyze data from a previous study in our lab to look at whether these representations are still present or whether they're different when people are making dietary decisions and are not explicitly instructed to think about the taste and health of the images. So this study was already published by Rosenblatt et al. In 2018. There were 85 participants and they completed 2 phases. The first of these was a rating phase where they just rated the taste and health of a series of snack food images. And then in the decision phase, they viewed a subset of these same images and they rated how much they wanted to consume each food instead. And this is what each trial in the decision phase looked like. So they would view the food image for 3 seconds, and then they would rate on a sliding scale similar to the one used in experiment 1, how much they wanted to eat each food at the end of the experiment. And there were 50 trials in this stage, and it all occurred in one block, while EEG was again recorded. So our analysis was basically the exact same approach as the one that we used in experiment 1. So we have similar figures here. These are the results for taste. But interestingly, the taste decoding was a little bit later in this experiment compared to experiment 1. So we can see that significant decoding started from about 750 milliseconds, but this particular window didn't remain significant after corrections for multiple comparisons. So the significant decoding that we have is in this window from 1100 milliseconds, which is quite a bit later than the decoding from experiment 1. And then when we look at the health decoding results, on the other hand, it was possible to decode health fairly earlier, so from a little bit after 500 milliseconds, which is about the same time as taste was decodable in experiment 1. And I'll talk a little bit later about why this may have been the case. But we also did one additional analysis in experiment 2, which was decoding the decision strength, which was the ratings of how much people wanted to eat each food at the end of the experiment. And we can see here that we have significant decoding a little bit after 800 milliseconds, which if we look at all the 3 graphs together for this analysis, the decision decoding was a little bit later than the decoding of both of these attributes. So what we might be seeing here is the integration of taste, even though this time window wasn't significant after corrections for multiple comparisons, but we might be seeing the integration of taste and health into these decisions if they're firstly processed and then integrated into the decision. Okay, so what does all this mean? Firstly, we found evidence that information on both taste and health is activated in the brain during the first second of dietary decision making. So previous studies had already kind of found evidence that taste information was present, but we found evidence that people also process health at this early stage. And this was also possible even when people were not explicitly instructed to think about taste and health. So this might be saying something about the way people make decisions in real life or how they process food cues in real life. Now, as I mentioned, taste was decodable earlier than health in experiment 1, but then this was kind of reversed in experiment 2. So I don't know exactly why this was, but I've put a couple of speculations here. So firstly, in experiment 2, the stimuli had a lot of variation in health, but not a lot of variation in taste, because the images were all snack foods, which were fairly appetizing, even the healthy ones, which were images of fruits and things like that. So people rated all of the stimuli as highly tasty. So it might be that because they saw all the images as being a similar taste level, they gave health processing greater priority. Another possibility is that because participants had already been exposed to the stimuli in the rating stage, their automatic craving reactions, which reflect taste value, may not have been as strong by the time they got to the decision phase. Whereas in experiment 1, they were viewing all of the stimuli for the first time. So for future studies, we might use these results to determine whether interventions might alter either these temporal processing dynamics or the strength of these representations of taste and health, so such as nutrition labeling or health warning labels. And thank you everyone for listening, and thank you to everyone who's been involved in this project.
Fantastic. Thank you, Elektra. And if you enjoyed the talk, please drop some praise in the chat, because we do not have any applause here. So we have time for a couple questions, which is great. The first question comes from Mohammed Abdelhak in the Q&A box, and he is asking, are the results in experiment 2 based on training from the data of experiment 1?
So that's a good question. No, the 2 experiments were completely independent of each other. So the training was based on data from experiment 2 completely. Great,
thanks. And there was also, Tyler is putting something in the chat here. Us panelists can't use that Q&A box. So Tyler is saying there's probably heaps of between subject and between trial variance at the later time points in the epoch. You might get better predictions if you can somehow realign the time points.
I think that's a really good suggestion. Yeah, I'm not really sure how to do that, but if it was possible to realign the time points, that would be really good. So maybe I'll talk to my supervisor about that, I guess. Thanks, Kyle.
Great. And you guys can also talk afterwards. And can I ask a very, very quick question before we close up? Unless there's another one that's coming from the audience, but it looks like you're training 3 different classifiers in experiment 2 or in study 2. Have you thought about taking the classifiers for taste and for health and applying them to the desirability to consume data and seeing which one might be able to kind of predict better, or when they can predict better, and how that might influence those choices?
No, I haven't done that, but I think that would be a really good idea. Maybe something to do in the future. We're still working on getting this paper written up, so it's not all finalized yet.
Sure.
That's a great.
Cool. Yeah, like a cross-decoding approach. That might be kind of interesting and informative. Great. Well, if there are no more questions for Elektra and for any of our panelists, we will go ahead and close up. But if the rest of my panelists might turn on their camera.